So let us start. I would like to introduce the first speaker, the first talk. We are going to have Professor John Tavis from the University of St. Louis in the US. And we are really glad that he is going to give us more the basic background about the therapeutic approaches for targeting, for target RNA degradation, so about the molecular background, how to use this strategy and for targeting HBV. Please, John, the stage is yours.
Thank you, Mara and Adam. I appreciate it. I particularly appreciate being invited to give a talk on something which I do not work on and have no knowledge of. So in any case, this will be a little bit creative. So feel free to laugh at me at frequent intervals. It's probably just a good thing in general. I want to start out by putting in a plug for ICE-HBV. ICE-HBV is a collection of the leading early stage translational and basic HBV researchers, and our goal is to advance cure for HBV using science-driven, thoughtful approaches to improve cure rates and eventually eliminate the virus. The important part on this slide is listed in bold down at the bottom. We are the voice for the HBV research community that is represented at this larger meeting that we've been at all week for scientific policy and public lobbying. So if you have an issue that you wish to be brought up and try and focus the science towards cure, please reach out to me. I'm easy to find on the web, and of course there is the connection. Our website is shown right there. Okay, so my topic is therapeutic approaches for targeted RNA degradation, background and strategies to target the virus. Okay, so let's start out with the basics. On the left you see here is the viral replication cycle. This audience doesn't need me to go into any details on it. The key parts here is this is the cccDNA. It is made by reverse transcription, and it is the template for all HBV RNAs that are involved in direct viral replication. There is one exception to that, is the HBsAg messenger RNA can also come from integrated genomes, and that is the predominant source of it in most e-antigen negative patients. And so all HBV RNAs are messenger RNAs, so it's not like some viruses where they segregate the functions of their roles. Each of the messenger RNAs each of the RNA is at least a messenger RNA. The pregenomic RNA, which is one of these longer forms of RNA that you see here, is also the RNA phase of the genome, so it is a dual function. Reducing HBV RNA levels obviously would reduce viral protein production, and if you reduce viral protein production and you reduce the RNA phase of the genome, well, obviously you're going to block viral replication too. And one of the nice things about HBV as far as these types of targeting agents goes is that all of the messenger RNAs are 3' coterminal, which means there's this region right here, if I remember correctly, it's around about 700 nucleotides, that if you target this with an RNA degrader or an RNA blocker, you're going to hit every single one of the viral RNAs, and so consequently this makes it a very logical drug target. And the pharmaceutical industry has not overlooked that obvious conclusion. So if you just think about, in general, the ways to reduce RNA accumulation in cells, you can do just as with all things. You either suppress production or increase degradation. That's the balance of the two always leads to the steady state accumulation level. So suppressing function is something that many people are approaching. And I just listed a number of the topics there that came to my mind. I'm sure there's more methods if you sit there and think longer. Epigenetic suppression of cccDNA, that's obviously a way to do it. You can target the transcription factors that lead to HBV messenger RNAs. You can induce aberrant splicing to produce dysfunctional RNAs. You could block RNA export from the nucleus. Those are all possible. And one of the ones that I find most exciting is the possibility of inhibiting or degrading the HBX protein, because that would reactivate the SMC5/6 silencing complex and use cellular mechanisms to shut down production of the RNAs. So that's pretty cool. On the degradation side of it, you could deliver an HBV-specific RNase, or if you deliver a DNase that targets the cccDNA, you'll get to the same spot. You could trigger a host-specific RNase. Okay, that's a good idea. But also the areas that appear to be a little bit more tractable pharmacologically are as you can use siRNA-induced degradation or antisense oligo-induced degradation or silencing. For the rest of today, we're primarily going to talk about these areas because those are the areas where we're starting to see some clinical data come through with this topic. Okay, so I'm going to briefly go over the mechanisms of these various modalities. Those of you who spend your lives working on this, go ahead and take a nap right now because this is going to be 101 level. In any case, siRNAs are exogenous double-stranded RNAs, and that when they get into the cell, they induce something called RNA interference, or RNAi. That's the general class of activities. RNAi is important in invertebrate, but not mammalian immune systems. siRNAs themselves are 21 to 23 nucleotide long duplex RNAs. They are usually heavily modified and conjugated to both promote stability and cellular uptake because the pharmacological parameters of bare RNA in the body are absolutely atrocious. RNA is the world's perfect substrate, and it really likes to get degraded by any enzyme that happens to blink at it. And then you've got, in these particular cases, you've got RNA-guided RNA recognition and RNA cleavage by the AGO2 cellular RNase that is found within the RISC complex. And you can see here a textbook-style description of this complex. It's important here to note that siRNAs can be highly specific due to the nucleotide complementarity between the guide strand, that's the RNA in the RISC complex, and the target RNA. So you can have a great deal of specificity there, and the beauty of it is it's, of course, plug and play because you can change that sequence to anything that you want. Let's move on to antisense oligos. They are also an RNA-based drug product. These are single-stranded and are typically longer than siRNAs. They're also, of course, modified heavily to promote stability and cellular uptake. There are multiple different ways of doing that, and those can be used to distinguish different drug products from different companies. Antisense oligos also bind to their target mRNAs by complementarity, leading to the possibility for very good specificity for the HBV RNAs. No, depending upon where they bind and a bunch of other things, they can have three mechanisms of action that have been reported. And so the first one that I want to report here is targeted degradation. The oligos are DNAs, and so if you bind them to an RNA, you get an RNA-DNA heteroduplex, which is, of course, the substrate of the RNase H1 classes of enzymes. And human RNase H1 is a common enzyme. It's essential for human life, and that will very promptly chew up the region that is targeted by the antisense oligo, destroying the RNA. Another possibility is that you can modulate splicing. If your antisense oligo happens to sit on a splice site or change the conformation of the RNA in a manner that the splice site is not recognized, you're going to get a different collection of RNAs coming out. And this process is being used in some cancer-based antisense oligo strategies. And finally, if you bind the antisense oligo to a spot where at the 5' end where the ribosome initially recognizes the RNA, you can block recognition of the RNA and directly suppress mRNA production. So those are the ways that antisense oligos can work. You got RNA destabilizers. Those are coming out. When I first heard about these a few years ago, I got really excited about them because I just thought it was mechanistically extraordinarily cool. And so what these do is there's been a bunch of work figuring out how the ones that are in clinical trials work. What they do is they target the proteins PADP5 or PADP7. These are non-canonical poly-A polymerases that can obviously add a poly-A tail to the end of a messenger RNA, but they intermix a few G residues in there, so it's not a pure string of adenines. That can helps to stabilize the RNA, And so consequently, if you inhibit those enzymes, you end up with a shorter poly-A that doesn't have those stabilizing Gs in there, and you can end up destabilizing the RNA post-transcriptionally. From the reading that I was able to do, DP5 and 7, at least conceptually appear to be fairly redundant, but there are differential suppression of the two enzymes by various molecules that are either in preclinical or clinical work. So this, again, I thought mechanistically was exceptionally cool. Okay, so now here I have to start out with a proactive apology to all of the industrial researchers in the room. I'm a preclinical mechanism kind of guy. I do not follow the clinical literature with a great deal of diligence. So I did the best I could to pull out some key examples from the publicly available data here. But during the question and answer period and the panel discussions, if I have slandered your companies, please correct me, okay? I do not intend to do that, but in any case, so again, I focused in on the post-treatment survival of the RNA. You know, how long are these things working after you stop giving the drug? And so I give a few examples here. The first one is with Vir Biotech's 2218 molecule. If you give that with interferon, up to 16% HBsAg loss, 24 weeks post-treatment, excuse me, in a phase 2 study, and development is ongoing with this molecule according to the publicly available data. Arbutus has got a molecule called 729. That, when treated, co-administered with a nucleoside analog, in one particular example, 7 of 9 patients that were followed for 72 weeks after treatment remained HBsAg negative, and with that positive result, of course, development is still ongoing. Janssen made a molecule called JNJ3989. If you administered that with nuke, you got up to 1 to 2 log HBs suppression 24 weeks post-treatment in a phase 2b trial. I found that completely fascinating when I learned about this, but for corporate reasons that I don't particularly understand, they have paused development of this molecule and are likely looking for a partner to outsource it to. Oligos has a molecule called 125-755. It's doing well in phase 1 monotherapy trials, safe, and it's got decent efficacy, but of course in a phase 1 trial you don't know much about a molecule yet. And then Roche and Dicerna have a molecule, I'm just going to call it 6346. It's in phase 3 monotherapies. Again, safety and efficacy are looking decent, and development appears to be progressing, again, from what I can gather from the publicly available information. There's an antisense oligo in the clinic. This is bepirovirsen. Okay, and you go ahead and laugh at me now. This would be a good cue for laughing at Tavis, okay? That is, when treated with a nuke, you got a 10% sustained HBsAg loss 24 weeks post-treatment in a phase 2 trial. And with that encouraging result, it's moving into phase 3 trials. And you've got the Arbutus AB161, which is a RNA destabilizer. It's in phase 1 trials, but I could find no publicly available data that has been released on it yet. Okay, so here's the conundrum that Adam brought up in the beginning, is that you can get durable messenger RNA suppression after antisense oligotherapy. And the suppression can be long-lived, but it can occur for a long period without complete HBs clearance. So 3989 plus nuke, remember you got a 1 to 2 log HBs suppression 24 weeks post-treatment, but that wasn't clearance, okay? So clearly that was not due to a functional cure, because the patients, can still be HBV DNA positive and HBs positive. So what's going on here? This is unusual. So, okay, so here's some possibility. All right, did we achieve a functional cure? In some of those patients, it looks like the answer is probably yes, and that's very good. We're all excited, yay. Perhaps we have a very long intracellular drug half-life. But 72 weeks is an awfully long time for a drug product to hang out in an active form in a cell. So I find that difficult to believe. So then you have to start thinking about cellular mechanisms. Are you inducing a persistent innate immune response? I don't know. Are you enhancing an adaptive immune response? That's possible. This should be testable quite easily. Are you restoring SMC56 silencing of the cccDNA? That's a little harder to test in a human being, of course, but it's certainly possible. So, you know, this is our conundrum, and this is what we're going to be exploring today. And I'm really looking forward to hearing discussions from people who actually know something about this topic and informing me as to what these mechanisms may or may not be. All right, thank you very much, and don't forget to go to the ICE-HBV website.
Thank you very much, John. I think as the introductory background, if there are urgent questions that you would like to ask John, otherwise, I think it's good that we discuss more later when we heard more from the different speakers. So thank you again.